Good morning, everyone. My apologies for being late. Welcome to the Advisory Committee on Plants and Animals of the Hawaii Department of Agriculture. This meeting is called to order on Friday, May 20th at 2022 at 9:04 a.m. according to my computer. My name is Darcy Oishi, chairperson of the committee. I have a memo dated May 13th, 2022 from Chairperson Shimabukuger Geyser designating me as the alternate to chair this committee should Dr. Helmut Rogg not be available, which he is not today. A copy of this memo will be filed in the meeting minutes for today. For those of you who do not know, I'm the acting manager for the Plant Pest Control Branch, and I've been a long-time alternate chair of this committee and sometimes chair of the committee. So good morning. I will disclose that I also have Stephanie Isley with CGAP's coordinating, CGAP's legal fellow here in the room with me. Rob, why don't you start off with our introductions?
Okay, thanks, Darcy. Rob Hoff, Department of Land and Natural Resources.
Mr. Eisen of ERP.
Yes, Tom Eisen with the Environmental Review Program of the Office of Planning and Sustainable Development, pinch hitting for the director of OPSD, Mary Alice Evans, who I believe is ex officio member of the committee.
Yes, Jody.
Sorry to jump in, but could everyone, when you introduce yourselves, could you also mention if there's anyone else in the room with you who's sent you?
Sorry
Nobody
about
here.
that. I'm alone.
I'm alone also.
Okay. Grace.
Good morning, everyone. Grace Simmons, Program Manager for the Department of Health Vector Control Branch, and recently assigned to sit in on the panel.
And I have a memo from Dr. Elizabeth Shard delegating Grace as ex officio representative to this committee. Letter is dated May 19th, 2022. I'll submit a copy to the meeting minutes. Thank you. Dr. Hawes.
Morning, I'm Maria Hawes. I'm with the University of Hawaii at Hilo, and I'm alone in this room.
And Mr. Matsui.
Ken Matsui, Petland Pets Pacifica. I'm here with my black lab.
Thank you. I will appreciate testimony from the lab at a later point. Looking forward to it. Okay, next on the agenda, we have approval of minutes for the meeting on December 10th, 2022. Did everyone have a chance to review the minutes?
I did. I had a minor correction, Darcy.
Go ahead, Rob.
Let's see if I can find it. Yeah, on page 8, the third complete paragraph, there's a typo. Mr. Hoff asked Mr. Oishi. He asked if only live plants were covered, not live plants.
Oh, I didn't catch that. I have a similar minor edit on page 6, second line. It should read facilities is risky, the intent, not the they intent. And then also on page 6, paragraph 3, which starts, Mr. Matsui asked Mr. Oishi, please strike the that in the sentence. Any other comments from the committee? Do I have a motion?
I move to approve the minutes as amended.
Second?
I second.
Okay, any discussion? Okay, any input from the public on the minutes? Okay, then I'll call for the vote. Chair votes aye. Rob?
Aye.
Tom,
Aye.
Grace,
Aye.
Maria,
Aye.
Ken.
Aye.
Great, motion carries. This is that was the easy vote. So agenda item 4, comments from the general public. At this point in time, we received 3 pieces of testimony from the Land and Natural Resources, Department of Land and Natural Resources. We did have it looks like some errors with our the hdOA.pq.testimony@hawaii.gov email address, where we're finding out testimony from the coordinating group on alien pest species and from Patrick Conant were not received. Members should have received copies of these testimonies. Any members of the public wishing to provide testimony, Kevin will explain to you now on how you can participate in this hybrid meeting. Thank you, Kevin.
Yeah, thank you, Darcy. So yes, for those in attendance from the public, if you are using Zoom, if you're connected to Zoom through a web browser or the app, just go ahead and click on the raise hand button next to your name. And then for those who are connected to Zoom via Zoom audio through the telephone, if you need to raise your hand, just click on #9. And if you need to mute or unmute yourself, type #6. So Darcy, I have Christy Martin with her hand raised up.
Okay.
Okay, so
Christy, I'm assuming you're going to provide testimony for some of the active submittals, and we can please confirm. You can let her in.
Christy, please state your name and affiliation for the record.
Aloha, Christy Martin with the Coordinating Group on Alien Pest Species, CGAPS.
Okay, and is it okay if I hear your testimony during the actual submittals you're interested in, Christy?
Yes, that's fine. Thank you.
Okay, thank you. Okay, if that's all, then we can proceed with the next item on our agenda, which is the submittals from Plant Quarantine. I'm assuming Chris is going to be reading our first 2 submittals. Or is that you, Jonathan?
Yeah, Chris will be doing the first 2, and then Noni will be doing the third one.
Okay, thank you. Mr. Kishimoto.
Hi, Chris, state your name and affiliation for the record.
Good morning, everyone. Chairperson Oishi and PNA members. My name is Chris Kishimoto. I'm with the Hawaii Department of Agriculture's Plant Quarantine Branch. I'd like to go over a request to number 1, preliminarily review the currently unlisted beetle Cyphrea uberabensis for future placement on the list of restricted animals Part A as a biocontrol agent for Tibucina herbacea and other related species in the family Melastomataceae by the United States Department of Agriculture's Forest Service. 2, to determine if the release of Cyphrea as a biocontrol agent of Tibucina herbacea and other related species in the family Melastomataceae by the United States Forest Service, the United States Department of Agriculture's Forest Service poses no significant impact on the environment. 3, provided that Cyphrea is placed on the list of restricted animals Part A, allow for its release from laboratory quarantine by permit for biocontrol of Tibucina and other related species in the family Melastomataceae by USDA's Forest Service. 4, provided that Cyphrea uberabensis is placed on the list of restricted animals Part A, allow for its importation and release by permit for biocontrol of Tibucina and other related species in the family Melastomataceae by the USDA's Forest Service. And 5, provided that Cyphrea uberabensis is placed on the list of restricted animals Part A, establish permit conditions for the importation and release of Cyphrea as a biocontrol agent of Tibucina and other related species in the family Melastomataceae. So for this particular request, the applicant and the importer, if a permit is issued, is going to be Dr. Tracy Johnson with the USDA Forest Service, Hawaii Volcanoes National Park Quarantine Facility located in Volcano, Hawaii. The applicant is requesting that the board place Cyphrea uberabensis on the list of restricted animals Part A for import and release for biocontrol of Tibucina herbacea and other related weed species in the family Melastomataceae. Cyphrea uberabensis was originally brought into Hawaii Volcanoes National Park's quarantine facility from Brazil in July of 2005 for biocontrol research and host range testing. In January of 2020, a draft environmental assessment was submitted to the Office of Environmental Quality Control with the anticipated finding of no significant impact. That draft was published in OEQC's environmental notice on January 23rd, 2020. So Cyphrea uberabensis is a small beetle that's native to Brazil and has been selected and evaluated as a new biocontrol agent for managing invasive melastome weeds in Hawaii. It is a narrowly host-specific leaf-feeding beetle intended for statewide field release to cause suppression of non-native plants in the genre Tibucina, Melastoma, which are both designated as noxious weeds, and Pterolepis. The beetle is expected to cause severe defoliation of targeted weeds without affecting any native or otherwise valued plants. Suppression of these weeds will benefit forest watersheds statewide. Initial quarantine will be at the USDA Forest Service Hawaii Volcanoes National Park Quarantine Facility at the Kilauea Research Station. The Cyphrea uberabensis colony will originate from insects collected from southern Brazil and shipped under a USDA Plant Protection and Quarantine permit to the Volcano Quarantine Facility for rearing and screening to eliminate associated natural enemies. Dr. Johnson will positively identify the insects and determine them to be free of natural enemies in preparation for release. Host specificity tests indicated that the host range of Cyphrea uberabensis is restricted to a few melastome species, all of which are non-native and all of which are considered invasive in Hawaii. Potential host preferences were evaluated on a total of 58 plant species in 30 different families. Testing revealed that Cyphrea uberabensis is narrowly host-specific within the family Melastomataceae and able to complete development on only 5 plant species, which are Tibucina herbacea, Tibucina longifolia, Pterolepis glomerata, Melastoma septemnervium, and Melastoma sanguineum. This request was submitted to the Advisory Subcommittee on Entomology for its review and recommendation. The Advisory Subcommittee recommendations and comments are as follows. So we sent this request to 6 of our members. Of the 6 members polled, 5 responded for the recommendation of future placement of Cyphrea uberabensis to the list of restricted animals Part A as a biocontrol agent against the noxious weed Tibucina herbacea and its related weed species in the family Melastomataceae. All 5 responding subcommittee members recommended approval. Number 2, on the question of whether or not the release of Cyphrea as a biocontrol agent of Tibucina and related weed species by USDA's Forest Service poses no significant negative impact on the environment. Again, all 5 responding subcommittee members unanimously agreed that, that there is no significant negative impact on the environment. On the question of whether or not to recommend approval or disapproval to allow the importation and release of Cyphrea by permit for biological control of Tibucina and related weed species, again, all 5 responding subcommittee members recommended approval. And lastly, for the recommendation to establish permit conditions for the import and release of Cyphrea, Again, all 5 subcommittee members that responded recommended approval. HDOA's Plant Pest Control Branch did have some concerns about the screening process and whether or not it should be done prior to release within the permit conditions. Also, whether or not Cyphrea could be released straight from quarantine on Hawaii Island, or whether or not it needed to be taken to Oahu for inspection. The plant quarantine branch consulted with our legal counsel, and it was determined that there is no requirement for Cyphrea to be transported back to Honolulu for inspection after the issuance of a permit. We've also amended permit conditions number 11 and number 5 in order to address these concerns from Plant Pest Control Branch and to comply with Chapter 150A-5. Thank you.
Okay, thank you, Chris. First off, I want to comment that we have gone out of order from the agenda. This should have been agenda item 2, not agenda item 1. Legal counsel, do I need to, since we deviated from the agenda, do I need to make a motion or anything to fix this?
Technically, to revise the agenda, you would have to get a motion, but I guess you can quickly do it if the
Okay.
members would.
I move that we adjust the agenda to review the biocontrol agent for Cyphrea uberensis to item number 1 for review, and instead of item number 2. Do I have a second? Thank you, Tom seconds. Any discussion or questions and comments from the public? Can I have the vote? Chair votes aye. Rob?
Aye.
Grace?
Aye.
Tom?
Aye.
Maria?
Aye.
And Ken?
Aye.
Motion carries. Thank you. Okay. I will, before I open up for discussion on this submittal, I want to state that I will be recusing myself from voting. I was notified that I would be chairing this meeting after I was sent and provided commentary for the Plant Pest Control Branch as the Subcommittee on Entomology. I would like to further disclose that this submittal, the Forest Service is a partner project for the Plant Pest Control Branch under an MOU between the USDA, Forest Service, and HDOA. Plant Pest Control Branch staff will actually be conducting the first releases consistent with those, the MOU between the 2 agencies. So as such, I'll be abstaining from voting. That said, any questions or concerns about my status?
I
Okay.
should warn something here. I expressed concern that I might have to recuse myself, but I did run it by the Ethics Commission, and T. Campbell said I probably do not have to recuse myself. The basis for concern is this beetle does attack the leaves of the Katapa plant. Katapa plant is found around Oahu, especially in Waimanalo. It surrounds the baseball field in Waimanalo Park. It also is in my friend's yard along the ocean in Waimanalo, and that tree protects her yard from being consumed by the ocean. Also, I use the Katapa leaves as a water conditioner. They have an exceptionally high tannic acid concentration, which makes water more close to the conditions of the Amazon basin. So fish like cardinals, discus, and neons do better with that in the water. But Keith said he doesn't think there is a problem. So I will probably vote, but I should disclose that I may have a problem.
Thank you, Ken. Do I have a motion?
Can we ask a couple of questions of PQ
Sure.
and your applicant?
Yes, of course.
I guess first up, maybe Mr. Kishimoto, PQ.
Chris? Oh,
Should I go ahead?
go ahead. Chris is in.
Okay. Chris, just a couple of questions on the conditions, starting with number 5 and the requirement to rear out 2 generations, which seems like a good safeguard for screening for parasitoids. Is that a standard condition across taxa? Or does that vary due to the biology? I'm just curious for, whether that's a standard condition or whether you're applying that specifically to this organism because of its biology.
It's usually a standard condition for us with things that like biocontrol agents and stuff like that that go into quarantine. But it also does vary on the biology. We try and pay close attention to if things are different for their life cycle, we'll try and alter it to reflect that if need be.
Right. Okay. Thank you. And then I also had a question on condition 1. And, the intention is that this project will eventually be implemented statewide. And I don't think anybody knows exactly how well this organism is going to disperse on its own, but there likely will need to be efforts to make sure that it gets to all of the populations of the target species. And the way that condition's worded is, as I interpret it, is that the applicant is going to have to do all of those releases and will not be able to hand it off to partners. And I'm wondering how that's managed as, clearly the initial releases will be conducted by Dr. Johnson, but as it becomes operational and becomes a management tool, what does an agency like DLNR or a nonprofit partner that might want to help with the control of this weed need to do to be able to acquire the agent and release it?
I think that's a good question. I think that at least for permit condition number 1, because HDOA is a partner agency with this, and they also have a quarantine facility of their own in Honolulu, that they can act as a partner agency and help with the release. As far as other partners like DLNR or other non-government agencies and stuff, I think we might have to ask our AG for clarification on that. It wasn't requested in this permit request, but it may be something that we can try and address before it gets to board.
Right. Just thinking down the road, as this becomes implemented, what we'll need to do, assuming this passes and the board and gets the approvals and is released, what's the eventual plan for implementation?
Yeah, I think definitely in the future, I guess when you can actually collect from the wild and stuff like that. I don't know how far into the future this would be, but once existing populations are at a level where they can be field collected and stuff, I think for sure at that point, partner agencies outside of USDA and HDOA wouldn't be prohibited from collecting and redistributing those things throughout the state.
Okay.
But HDOA does have facilities statewide on all the islands that definitely initial releases can be done, I think, by HDOA staff.
In the past,
Could I add, reading the
oh, go
condition?
ahead, Tom.
The condition says that that transfer can be approved by the board, so it can happen, just the board would need to allow for it either at the future point or maybe as part of the approval now, they could factor that into their decision to allow the transfer to these other entities.
I think the committee and the board needs to look at it for the permit application is for actually the introduction, the actual release into the environment. So those releases will be conducted both by the Forest Service and HDOA Plant Pest Control once it's established, because the intent of this application is for field release into the natural environment. So the permit is only governing the activities up until establishment. That is the traditional interpretation of this permit condition for our prior biocontrol releases.
Once it becomes established, doesn't it become conditionally approved?
No, it would always be restricted.
So what that functionally means is any new introductions of other material would have to go through a similar process, Ken. Not anyone can just bring it in on their own.
So if I pick a leaf that has a beetle on it, like a Katapa leaf, I can't use that leaf. I have to leave it as it is.
Once it's considered established, no, you can do, it could be moved. If you're taking it from a natural population, it can be moved around. That's at least my interpretation, and legal counsel can correct me if I'm incorrect.
Darcy, I have Jonathan raising his hand.
Jonathan?
Yeah, so Jonathan Ho, Inspection and Compliance Section Chief for the Plant Quarantine Branch. So yeah, so Chris is correct. The placement will be for restricted A and the intent is for release. However, once it becomes established, it is still restricted. So as Darcy said, importation is still restricted. However, individual possession, while technically letter of the law is prohibited because it is a restricted organism, the branch doesn't take action against things that are established and widespread. Because, for example, geckos, all gecko species are on the essentially on the restricted A list or prohibited. We don't go after people who have a house gecko as a pet. You know, that's essentially how the intent of the law is set up. You know, obviously it's to manage things that are controllable. And obviously if the intent is to introduce and spread, we obviously want to control how that's being done. And once it's done, the movement and the possession within the state is essentially unregulated. I hope that helps, Ken.
Yeah. That's helpful, Jonathan.
Oh, I think we lost him.
Uh-oh.
Maybe I'll repeat myself when he comes back. But yeah, so I mean, for everybody else, I think that's kind of how the intent is. I mean, you put it on this high restrictive level, and obviously once the intent is there to you know, to distribute, you know, obviously at the agencies whose mission is to do that work, and then once it's established all over the place, it's, you know, it's kind of free to do what it needs to do.
The permit condition also relates to what Dr. Johnson can do with it prior to establishment, which is he cannot give it or transfer it or conduct any of these activities without express permission from the board.
Correct.
Do I have any other?
I don't know if other committee members had more questions, but I was going to ask questions of the applicant as well, if possible. Darcy, you're muted.
Thank you. Kevin, please let Tracy Johnson into the room, please.
Tracy, please state your name and affiliation for the record.
Hi, this is Tracy Johnson. I'm a research entomologist with the USDA Forest Service Pacific Southwest Research Station, Institute of Pacific Islands Forestry.
Morning, Dr. Johnson.
Can you hear me?
Yeah,
Sorry.
hear you loud and clear.
Okay, go.
Great. I just wanted to, ask you whether you felt the permit conditions were workable for implementing the project. They sound, as we've discussed them, they sound like, reasonable precautions to take. And I think PQ has done a good job, but I want to make sure that this project is also workable so it's implemented, and would ask if you have any comments on the conditions as they're currently stated.
I don't think I have any problems. I'm not sure that I've seen exactly what they are, but I think it was rearing a couple of generations before release.
2 generations.
There was the corrections or adjustments in terms of whether it needed to go through Honolulu and stuff. I wasn't totally clear on that either, but it sounds like those have been resolved. So yeah, I guess the short answer is no, I don't think I have any problems with it.
Okay. And then I wasn't quite sure what Ken was referring to.
Yeah.
Katapa in the Bignoniaceae is.
Katapa is it's a plant in the Combretaceae,
Okay.
and that is a common coastal tree. Everybody sees it at the beach, but that's not a viable host plant for this insect. In the testing, there was some really minor feeding, and this is common in our testing when we put the insects on plants and don't give them anything else to eat. Sometimes they take a few nibbles of something that otherwise isn't edible to them. So when we put the insects on the plants for an extended period and force them to try to survive, they can't survive on Katapa. So it's not going to be a host plant in real life. So yeah, I don't think there's any real reason to be worried about any impacts on that.
Thanks, Tracy. I don't have any questions further.
Do we have any other questions from the committee for the applicant? Or Mr. Kishimoto? Ken, are you still connected by audio through the phone? Okay. I see a 677. I see a 677 number. I'm not sure if that's Ken. Ken, is that you?
That's me.
Okay, great. Ken, do you have any questions or concerns?
To what extent will the leaves of Katapa trees be removed
Tracy, Kevin, could you let Tracy Johnson back into the room, please?
Hi, can you hear me?
Yes.
So our expectation is zero removal of any leaf material by this insect on Katapa.
And my
And why
understanding,
did the
Tracy,
report show that it did?
So the report shows that when you put it in a small cage or container with a variety of different plants, sometimes the insects will take a bite to figure out if they can eat it. And so you do get damage, and that's common in all of these kinds of testing. But the damage isn't sustained, so they don't recognize it as a plant that they can eat, and they don't survive on it to be able to live a long time and reproduce, lay their eggs and all that. So
Well, no, that's not the question whether or not the life cycle of the beetle will continue on it. But the question is, to what extent will the Katapa plants be decimated by their absence of leaves because the beetles are eating them?
yeah, so basically what I'm saying is the beetles are not going to eat Katapa at all.
But that's not what the research showed.
Ken, basically we have, what we have is a no choice test. This is what it's called. And a no choice test basically involves, will it feed on it or will it die? In some of these tests, sometimes what happens is the organism will taste basically it will taste the leaves, and then, so that counts as feeding, but it will not actually be able to carry out its life cycle. It will not continuously feed. So we have feeding damage, but it is not impactful to the plant. And this likely would not happen in a setting where the organism is forced to stay there. It can wander around, it can look for appropriate food for it to feed on. And this is also on the assumption that the adults are going to be forced to lay their eggs in or on the plant that's being affected. So in the natural environment, Cyphrea would be laying its eggs closer to proper hosts and not necessarily Katapa.
Yeah, I'd like to I'll point out to help alleviate Ken's concerns that the range of the target plant doesn't overlap with Catalpa either. So you're not going to find those two plants together anyway.
Okay, thank you.
Any other questions and concerns? Is there anyone who would like to make a motion?
I'm happy to try to give a motion. There's a lot of parts of the submittal, and so I'm wondering if I can, if it's acceptable to make a motion to recommend to the board approval of requests 1 through 5 for the application submitted by USDA.
I have no problem with that. Legal counsel? Is that satisfactory?
That's fine.
Okay. Do I have a second?
Second.
Second.
Darcy,
Yes, Jonathan.
do you want to give public testimony?
I was going to ask for comments after the second. So I heard Maria and Ken say second. Is that correct? So I heard Maria first. So Ken,
That's fine, doesn't
sorry.
matter.
Do I have any comments or questions from the public? Christy,
Looking for my unmute button. Thank you so much.
you found the unmute button.
Thank you. Mahalo for this opportunity to provide testimony. Christy Martin with the Coordinating Group on Alien Pest Species. CGAPS is in strong support of this proposal. Biocontrol has gone through a lot of changes, and we think Dr. Johnson's work on this has been stellar. He has clearly worked on this for a number of years and has done all of the tests needed to be able to show that this is going to cause the intended impacts on Tibicina while causing no impacts to other species. And we just appreciate the work and urge you to approve this. I just wanted to relay also, I was listening to a biocontrol webinar the other day, and the way the practitioner put it is that this is actually biocontrol, it's not bioeradication. And so the Tibicina and other related species are going to continue to exist in the forest, just at a lower and less vigorous level. And that will give our native species some chance to compete. Mahalo.
Do I have any other public testimony? Do I have any additional questions or comments from the committee? There being none, I'll call for the vote. As I stated above, I will be abstaining. Rob,
Aye.
Grace,
Aye.
Tom,
Aye.
Dr. Haas,
Aye.
Ken.
Aye.
Okay, motion carries. Thank you. We are up for our next submittal from Plant Quarantine. Mr. Kishimoto, if you could be let back into the room by Kevin. And
Dorothy, I'm having problems connecting on the visual. I don't know why.
yes, we're aware, but as long as you can hear, we can proceed.
Okay.
Okay, and we can hear you just fine, Ken.
Okay.
Okay, hi guys. Sorry about getting my mix up there
That's
with
okay.
the submittals. Apologize for that. But the second request is to preliminarily review the currently unlisted moth, Eucalacia chrysippe, for future placement on the list of restricted animals Part A as a biocontrol agent of Myconia calvescens by the United States Department of Agriculture's Forest Service. Number two, to determine if the release of Eucalacia chrysippe as a biocontrol agent of Myconia by the USDA Forest Service poses no significant impact on the environment. Three, provided that Eucalacia chrysippe is placed on the list of restricted animals Part A, allow for its release from laboratory quarantine by permit for biocontrol of Myconia by USDA's Forest Service. Four, provided that Eucalacia chrysippe is placed on the list of restricted animals Part A, allow for its import and release by permit for biocontrol of Myconia by the USDA's Forest Service. And number five, provided that Eucalacia chrysippe is placed on the list of restricted animals Part A to establish permit conditions for the import and release as a biocontrol agent of Myconia calvescens by USDA's Forest Service. Once again, the importer and the applicant for this request is Dr. Tracy Johnson from the Institute of Pacific Island Forestry, USDA Forest Service, Pacific Southwest Research Station located in Volcano, Hawaii. Dr. Johnson is requesting that the board place Eucalacia chrysippe on the list of restricted animals Part A for import and release of biocontrol as a biocontrol agent of Myconia calvescens, a state listed noxious weed. Chrysippe was originally brought into Hawaii Volcanoes National Park's quarantine facility from Costa Rica in 2012 for biocontrol research and host range testing. In April of 2020, a draft environmental assessment was submitted to the Office of Environmental Quality Control with an anticipated finding of no significant impact. That draft was published in OEQC's environmental notice on April 23rd, 2020. The Hawaii Department of Agriculture and the Hawaii Department of Land and Natural Resources propose the field release on state lands in Hawaii of Eucalacea chrysippe, the golden summerback butterfly, for biological control of Myconia calvescens, a state-listed noxious weed. Myconia is considered one of Hawaii's most invasive plants, whose exceptionally large leaves shade out and outcompete other species, effectively forming a monoculture. Uncontrolled growth can overwhelm highly diverse native wet forest ecosystems that are home to critically endangered species and essential to our freshwater resources. Despite major efforts using chemical control, the species continues to proliferate, particularly on Maui and Hawaii Islands. E. chrysippe is a natural herbivore of Myconia in its native range, whose larvae feed in large numbers on Myconia leaves. Extensive testing has shown E. chrysippe to be host-specific on Myconia and other closely related members of the Melastomataceae family, all of which are non-native weeds in Hawaii. Because E. chrysippe is limited to feeding on a small pool of closely related non-native weeds, and with its potential to provide control on Myconia, its release is expected to be beneficial to the state's forest and hydrology, and adverse effects are expected to be negligible. Host specificity tests and observations in the insect's native range clearly demonstrate that E. chrysippe is host specific to a narrow subset of plants in the family Melastomataceae, all of which are invasive to Hawaii. Feeding by E. chrysippe is expected to reduce foliage and suppress vigor of Myconia trees, allowing other species to persist and compete, to the long-term benefit of Hawaii's forests and watersheds. Host specificity tests with larvae of E. chrysippe were conducted from 2012 to 2014 in laboratories in Hawaii at the Hawaii Volcanoes National Park Quarantine Facility and in Costa Rica. An emphasis was placed on testing plants in the order Myrtales, specifically on species within the families Melastomataceae, Myrtaceae, Combretaceae, Lathraceae, and Onagraceae. In addition, species from more distantly related taxa, but with economic, cultural, and/or ecological significance in Hawaii, were selected based on input from the U.S. Fish and Wildlife Service, consultations with members of the agricultural community, and expert sources on native Hawaiian plants. In total, 73 species of plants from 19 families were examined for suitability as hosts for E. chrysippe. Results of host specificity studies showed that among the 73 species tested, E. chrysippe larvae feed on and survive primarily on Myconia calvescens and a few closely related species within the tribe Myconiae. This request was submitted to the Advisory Subcommittee on Entomology for its review and recommendation. Advisory Subcommittee recommendations and comments are as follows. Again, of the 6 subcommittee members polled, only 5 responded. So for question number 1 on whether to recommend approval or disapproval of future list placement of Eucalacea chrysippe on the list of restricted animals, Part A, as a biocontrol agent of Myconia calvescens, the 5 responding subcommittee members all unanimously recommended approval. Number 2, on the question of whether the release of Eucalacea chrysippe as a biocontrol agent of Myconia by USDA's Forest Service poses no significant impact on the environment, all 5 responding subcommittee members agreed that there is no significant impact to the environment. For question number 3, whether or not to recommend approval or disapproval to allow the import and release of E. chrysippe by permit for biological control of Myconia by USDA's Forest Service, All 5 responding subcommittee members approved as well, recommended approval as well. Number 4, for the recommendation to establish permit conditions for the import and release of Eucalacea as a biocontrol agent of Myconia. Again, all 5 subcommittee members that responded recommended approval. The Plant Pest Control branch of the Hawaii Department of Agriculture basically had the same concerns as with the previous recommendation. I mean, with the previous submittal, about screening upon import, as well as whether or not the organisms needed to be transported to Honolulu for inspection after a permit has been granted. Once again, the plant quarantine branch consulted with legal counsel, and it was determined that there would be no requirement for E. crypsipy to be transported to Honolulu for inspection after a permit is issued. And also, we've altered and amended permit conditions number 5 and number 11 to reflect this and to comply with Chapter 150A.
Thank you, Chris. As with the prior submittal, I just wanted to disclose that I was notified of this after I had already supplied comments to the as part of the subcommittee on entomology, and as such, I will be recusing myself from voting. And similarly, this project is also a partner project between the Hawaii Department of Agriculture's Plant Pest Control Branch and the U.S. Forest Service. Adding additional reasons for my recusal. Do I have any questions or concerns from the committee that can be presented to Mr. Kishimoto or Dr. Johnson? Wow, this is unusual. Any comments or support from the general public?
Darcy, I have Christy Martin with her hand
Christy,
raised.
please let Christy in, please.
Aloha Chair, members of the committee, mahalo. Christy Martin, Coordinating Group on Alien Pest Species. Again, we would like to voice our strong support for this proposal. We loved visiting the facility, Dr. Johnson's facility, to see the work going on there. We were able to see these Myconia butterfly in captivity, and we are excited to see it out on the landscape. As you folks know, invasive species are one of the greatest threats we have to our economy, natural environment, and the health and lifestyle of our people, us. Invasive species are also the greatest driver of biodiversity loss in Hawaii. And this is a great step towards addressing one of the big invaders of our wet forests, Myconia. So thank you. We appreciate your support for this proposal.
I should have asked, Dr. Johnson, did you have anything that you would like to tell the committee?
Hi, can you hear me?
Yes.
I not really. I mean, I'm here to respond to questions if anyone has any questions.
Okay, great. Thank you.
I do have one question. How do you select the plants to be tested? Katapa was tested on the beetle, but not tested on this insect. Why?
Let's see. I thought it was included, but yeah, I mean, we just try to get a kind of representative samples of diverse plants that, depending on the target species, the target plants, and it's how diverse its relatives are, and then we select kind of representative non-relatives. And yeah, I don't remember whether Katapa was ever tested.
I see it on the list.
Is it on the list?
I'm on the Myconia submittal PDF page 140, or no, sorry, PDF page 125, Convertaceae terminalia Katapa.
Yeah, I thought it was tested.
Okay,
But
I'm sorry, I didn't see it.
I would
See it,
have to
Ben?
go back and look through all the detailed stuff. But yeah, so why we select different, we can't test everything. It's not realistic to test all plants, all 2,000 or 3,000 species of plants that are occurring in Hawaii. So we try to get things that are representative.
Okay,
And again, Dr. Johnson, are you okay with the permit conditions as they stand as far as being able to carry out them all?
I'm not, I don't have them like the detailed conditions in front of me, so I'm not sure exactly what they specify. This insect though it's more difficult to rear through its life cycle. So for example, our quarantine, we can't easily rear it through its complete life cycle inside our quarantine. So the plan for it is to import eggs from Costa Rica and then grow them up as larvae, get them to mature, and then introduce them into the environment as adults. So that would be the process. And so we would be screening along all of those stages, basically examining and making sure there's no natural enemies present.
Mr.
Right.
Kishimoto, how does that impact the permit condition for?
Yeah. What is that, 5?
Yeah, the screening of predators, parasites, paratoids on permit condition 5. Since Dr. Johnson is basically saying he will not be able to execute rearing for 2 generations.
Yeah, so if this is something that can be amended, if it's okay with the committee, and we can definitely recommend that to the board as well with a notation that it's very difficult to rear through its full life cycle.
Dr. Johnson, is it possible to carry out 2 generations in containment?
Am I still live?
Yes.
Okay. Not in our current facility, at least in the ways we've tried so far. So basically my plan would be to release it into the environment inside a large field cage, a very large field cage. But that's outside containment. So that's the way I would manage kind of propagating it, but it would be outside of containment.
And so as far as monitoring parasites and parasitoids and the different life cycles as you rear them out from eggs, are you confident that you'll be able to detect any other organisms in that?
Yeah. So we're familiar with the organisms, the natural enemies from Costa Rica. And there are some in the eggs, there's some in the larvae, there's some in the pupae. And we can monitor for those along every stage of the life cycle. So that's not a problem.
This question is for Chris then. A large field cage could be read as an approved PQB containment facility, correct?
That's a pretty good question. I would assume that as long as Dr. Johnson can adequately make sure that containment, doesn't get breached, and there's proper protocols in place, I would assume that it would be agreeable with PQ.
Okay, so then modifications could be made to the permit condition 5, because the USDA approved insect containment facility would actually be referencing what's actually included within our USDA permits, both Plant Pest Control Branch and the Forest Service, which is our brick and mortar containment facilities. But if we could allow screening in a outdoor facility, that would facilitate us being able to meet the permit conditions without breaking, while still allowing for screening for parasites, predators, and hypers.
Yeah, I think we can agree to that. You know, as long as we have the understanding like that, you know, this is not really like, you know, what's going to be like a consistent practice or something consistent. It's based on a case-by-case basis on the organism and its biology. And also if it's, you know, if it's specifically laid out what the changes are going to be, so we can put that into the permit conditions to adequately reflect that.
Dr. Johnson, have you found, have any of these, of the known predators, parasites, and hypers been found in your testing and evaluation processes here?
Let's see here. Yeah, I mean, we do, because if we bring in eggs, sometimes we'll find natural enemies in the eggs. So we screen them out immediately. Larvae, I think we've only identified the pests, the natural enemies of the larvae and the pupae from Costa Rica, where they're kind of exposed in the environment. Because when we bring in the eggs, there's not yet a chance for those natural enemies to attack the insect. So I don't think there are any that, so what would have to happen would be the natural enemy would have to lay its egg inside the egg, and then the parasite would have to emerge from a larva. There are egg larval parasitoids, but I don't think we've identified any from this butterfly ever. But we would be watching for that. So we would definitely screen for that inside our maximum containment first before we released it into an outdoor large cage containment.
Okay, regarding the large outdoor cage, so you're going to be looking for other foods that may complete the life cycle of the butterfly. And if you find that it is another plant that may be hurt, would you reconsider, or is the political pressure so great that you would release anyway?
Let's see. So the testing has been completed already. The screening we're talking about after bringing this in prior to release is screening for natural enemies on the butterfly. Sorry. We're not proposing to do additional testing of host plants beyond the testing we've already done. And all the testing we've done so far indicates that it's just the melostomes, and specifically for this insect, there's only 2 species in Hawaii that we expect it to feed on and reproduce on. And those are Myconia calvescens and Tetrazygia bicolor, which more recently has been revised and thrown into the same genus. So it's only Myconia species that this insect is going to eat, based on all of our
If you're
testing.
looking for just diseases or other parasites, why do you need a larger enclosure? It would seem harder to spot the butterflies and their offspring in a larger enclosure with multiple holes.
Yeah, so we don't need the large enclosure to screen for the natural enemies, actually, but we do need a large enclosure if we're going to propagate the insect under a containment and under basically in a situation where we can mass produce it in the state. We would have to have it inside of a cage. So that's the reason for having a big caged area to release it in initially, so that we can have it lay its eggs on Myconia trees that are under a big cage, and we can go revisit, collect those eggs and propagate them and like transfer them to from the Big Island to Maui. So it's because this insect, so these butterflies need large spaces to reproduce. They fly around, right? And their behavior requires a very large space and particular lighting
So it's a
conditions.
space and not necessarily a missing food element.
Yeah, no, it's not about the food, it's about the space.
Okay.
So Chris, would we be able to amend condition 5 to remove the 2 generations to inspecting through the various life stages and continued inspection in the outdoor containment? Would that solve this issue?
Yeah, I think we can do that. As long as the committee kind of specifically puts down, I guess how they want that language in permit condition number 5 to read. That'll help us out a lot. Also, I wanted to ask Dr. Johnson if he would be okay with us just allowing the import of eggs and no other stages of development, just because eggs seem to be the least risky of the life stages to import.
Yeah, that's fine. That's realistically, that's what we do. So I'm fine with that as a restriction.
Okay, thank you.
Great. So would that also, what condition, or no, that would just be, would any condition have to change for that, Chris? I think that sounds like a good idea. I'm just trying to see where we would want to insert that.
We can either put it in condition number 1. We just put like the restricted articles, comma, eggs of Eucalacea chrysippe, or we can make that into an entirely new permit condition, maybe into permit condition number 2, whatever works best for the committee and the board.
Maybe just simply saying only eggs will be permitted in a additional permit condition. I think that might be the clearest and easiest way to do that. Yeah.
Okay.
Is that what other committee members are thinking too? I see nods, maybe.
I agree.
Okay.
Yeah, we can put that into maybe like permit condition number 2. So it's right in the beginning.
Yeah. And then did you want our help in language for 5 beyond what I suggested?
Yeah, I would appreciate that.
Sure.
I mean, I kind of know what you guys want, but maybe putting it into words is a little bit tricky sometimes.
So I guess what I would suggest that I'll give it a try. Upon entry into PQB approved containment facilities, the restricted articles shall be screened for other species, predators, parasites, parasitoids, or hyperparasitoids throughout the various stages of the life cycle. further monitoring for these organisms will occur when the butterfly has been taken to an outdoor containment to be mass reared. Does other committee members, does that cover what we just our discussion? Oh, Jonathan has his hand up.
Jonathan?
Let me, okay, my hand went down. Yeah, so Jonathan Ho, Inspection and Compliance Section Chief. I'm kind of good at wordsmithing, so maybe I can help. So I think obviously for that particular condition, we need to remove for a minimum of 2 generations. So I think if we we strike the 2 generations, and then maybe we got to figure out, I guess, that maybe obviously somebody more technical. I think up unto a butterfly, right? That has to occur, right? I mean, the restricted articles shall be screened until they complete their life cycle, and I don't know whatever the term is, in these 2 areas, because right, we want that in containment. And then I think I think either another condition or add into this, should no species, predators, parasites, or whatever be found, the restricted articles may be transferred to a PQB approved, I forgot what the term is, a rearing facility located at however we want to define it. So it's very clear that, they got to go to containment. I think that the containment facility is the facilities themselves, the brick and mortar. They're going to complete, from egg to butterfly. And then, provided there are no pests or parasitoids found, it can then be transferred, because they're tied to each other, one and then the other. So I think, striking the 2 generations, whatever we want to call up until a butterfly, or till their maturity, up to a maturity or reproductive state, or whatever we want to call it, in those 2 places, provided no, predators, parasites, parasitoids, hyperparasitoids are found, can be placed into a PQB approved rearing site for production or something like that. And then, so I think the second thing is condition 6 needs to be addressed, because condition 6 basically says if it's parasitoids, they got to destroy everything. And clearly that's not what Dr. Johnson is doing. Obviously his ability to screen throughout the process is there. And I think we just gonna have to, continue, or so maybe like we're gonna have to, instead of destroying the entire lot, it's gonna have to be destroying the infested restricted articles. And then, should the adults potentially be infested, maybe then you do destruction. I'm not sure if that's something that Dr. Johnson would be doing, but I think that condition kind of contradicts, I think, I guess the process that he's being provided.
Mm-hmm. Right. Did you have any comments on that, Tracy?
No, that sounds all reasonable. We actually do, inside a containment facility, the insects are subcontained in little containers. So each egg mass would be in its own separate space, separated. So any natural enemies that might emerge from one egg mass wouldn't be associated with another egg mass. So we subdivide everything carefully along the way. And so that the larvae would be the same, they'd be on separate plants. But yeah, if it came to the adults and we found natural enemies in adults, at that point, yeah, we'd probably say, oh, okay, hold on, we're not going to be able to release any of these adults, and we'd back up. So, but yeah, there aren't any natural enemies that I know of. that would fit that description.
My only issue is that is an extremely wordy permit condition, Jonathan. So can we streamline it a bit to reflect something like, I mean, basically requiring it to go into approved facilities, PQB approved facilities, right? Following a process and a screening and approved SOP process, reviewed and approved by the branch. Because it's in this case, containment of some sort is important, but I think the process by which Dr. Johnson is following is actually more important than in terms of ensuring that everything is not parasitized or hyperparasitized.
Correct. And I think, you know, obviously we don't want to prevent Dr. Johnson from doing what he needs to do, making the condition so restrictive that he can't deviate from it. But at the same time, obviously we want the safeguard, you know, I think in the generalist of senses, because I think I won't speak for the committee, but I think the committee does want to ensure that, you know, there are no associated, you know, parasitoids and things like that. And I think what Dr. Johnson is providing it is something I think that is acceptable. And I think having the condition be a little wordy isn't really gonna, I think, really affect anything. And I think,
well,
I think it's,
as long as it's clear.
Yes,
I think
no,
what I want to see, Jonathan, is that there is an SOP process that's clearly spelled out that can accommodate both the safety and needs of this committee and the board, while still allowing flow for Dr. Johnson to actually execute these releases in a safe, productive manner.
yeah, so condition 18 has that he has to provide a biosecurity manual. So it's kind of
a
already,
manual is separate from is different from I mean,
because it talks about introduction of pests and accidental escape and spread of diseases and pests that are associated with it. And again, it's a general term that we, you know, it's kind of a standard condition. That's a general term that's really designed to be applicable to each request, because obviously we don't want to have to keep rewriting that particular request for every single import that happens. And obviously, you know, we can work with those applicants, you know, based off of, you know, their particular scenario and situation to manage that the manual. I mean, what we call quote unquote a manual. But again, it's something that, you know, everybody agrees upon as being done. And, you know, and obviously, you know, it's not something we necessarily want to post, because then everybody will know what they're doing. And if you have security and things like that in there, we don't want people to, you know what their security is and break into the places and stuff like that. But that's kind of how we've been doing it. I think what Dr. Johnson is going to do is, I don't think so much the issue. I think it's just how are we going to word it so that he can get a permit to do what he needs to do long term.
Would you like me to take a shot at condition 6? Or do you guys got what you need? If you guys have what you need, I mean, I feel like I understand what Dr. Johnson's going to do, and I am comfortable with the protocols that we've discussed. But if you feel like you need us to articulate that more, I'm happy to give it a try.
So I guess to me, I personally don't think that Chris or I, you know, Chris can shake his head if he disagrees with me, but I don't believe that Chris or I have any misconception in terms of what the committee would like to see the conditions look like. And I think in the motion, amending condition 5 to, to insert language to allow for the importation of eggs to adults in the containment facility and accommodating the use of a rearing, whatever that containment rearing thing, and then to adjust condition 6 to, to meet Dr. Johnson's current screening process. I think clearly has the intent of the, and obviously adjusting condition 1 for the eggs. I think just putting that general comment into the motion, we can work on the actual conditions, which I don't think is very critical, and clearly it'll meet the intent. And obviously, Jody's here. She'll make sure that we are hitting those check marks. But I don't think we need the actual verbiage, because I think it's crystal clear what the committee wants. That being said, if you guys want to have language specifically, I can try to work on something and maybe put it in the text or something where everybody can see it and you guys can read it off if that's what you'd like. Either way, it's fine.
I'm comfortable with discussing, including the intent in the motion. Just for clarification, Jonathan, if something needed to be tweaked when this went before the board, would there be the ability to do that then without having to bring it back to this committee if there was a minor detail or something that the Board of Agriculture wanted to further clarify?
Yeah, absolutely.
Okay.
That's the whole intent of the process, right?
Great.
The conditions are kind of living and breathing until the board ultimately approves them, and that's when they're kind of set in stone.
Yep.
So,
Okay.
yeah, if somebody sees something, testimony can be included, or we can work with it. Obviously, we'll put a PQB note and to adjust any changes that should arise after the fact.
Okay.
How about we've been going at it for an hour and a half. How about we take a break before we call for a vote to allow some drafting, well, necessary bodily functions that may need to be addressed, but also to allow some time for drafting of comments and a motion before we vote. Is that acceptable to the committee?
Sure.
Okay, so how about we take, Jonathan, I know you just turned off your camera. How much time would you like?
Yeah, no, if you guys,
A 5-minute break
I think 5 minutes,
or-
yeah,
Okay.
I'll come up with something and then I'll just post it in the chat. I won't, well, I don't know if I can do the chat with track changes or something, but maybe I can just redo the conditions and as verbatim what I think everybody wants.
Okay, so at 10:27, we'll be taking a 5-minute break. Thank you.
Okay.
It is 10:30. What happened to my time on my computer? 10:33. Is the committee ready to reconvene? Rob, next time you need to DoorDash your snacks to us too.
I'm ready.
Okay. 10:34, this committee is reconvened. I hope everyone is feeling a bit more refreshed. So do we have any modifications for permit conditions, or do we have a motion for us to proceed?
Hey, Chris, did they want to present some language, or should I take a stab at making a motion?
Yeah, Jonathan and Chris, do you have any, were you able to take a stab at modifying some permit conditions?
I was not able to, but I think Jonathan is trying to work on something right now. He might post it in the comments or in the chat section if you can't come up with it right away. But yeah, you guys are welcome to come up with a motion, I think, if you want.
Okay. Well, I guess I'll give it a stab. So I'd like to move to recommend the Board of Agriculture approve items 1 through 5 on the application from USDA for biocontrol of Myconia. I should specify since there's 2, with amendments to the conditions, including the addition of a condition to require only eggs will be imported, to modify condition 5 by removing the requirement of rearing 2 generations and inserting that all life stages will be inspected for hyperparasitoids, predators, etc. And an amendment to condition 6 that specifies that egg masses will be screened individually and any infected egg masses will be discarded and destroyed, while uninfected ones will be acceptable to rear out to adults. Any predators or parasitoids found in later life stages will result in destruction of the colony.
Do I have a second?
A second.
Thank you, Dr. Hawes. Any discussion on the motion? I would like, having said any discussion on the motion, I have a point. For permit condition 5, Mr. Kishimoto, if you could add in imported eggs to distinguish the imported eggs from any that Dr. Johnson produces within the containment facility or in-field cages, etc.
Yes, we can do that.
Okay. Any other discussion? There being any questions or concerns from the applicant or the audience? There being none, let's have the vote. As I stated earlier, I am abstaining from voting on this one. Since you're up on my screen first, Maria.
Aye.
Oh, wait, was there a second?
Yeah, there needs to be a second.
Oh, I thought Dr. Hawes seconded it.
I did second it.
Yeah.
My apologies, sorry.
All right. So motion by Mr. Hoff, seconded by Dr. Hawes. And Dr. Hawes has just voted aye. Rob?
Aye.
Grace?
Aye.
Tom?
Aye.
Ken?
Aye.
Motion carries. Thank you.
Thank you, everyone.
Thank you, Mr. Kishimoto. Thank you, Dr. Johnson. We have our next agenda item for importation of penguins. Ms. Putnam, please.
Hi, Noni, state your name and affiliation for the record.
Noni, you're muted.
Okay. Good morning, everyone. Can everybody hear me?
Yes. Noni, could you state your name and affiliation for the record?
That's up next, working on that. So,
Thank you.
good morning, Chair and members of the Advisory Committee on Plants and Animals. My name is Noni Putnam, and I am the Land Vertebrate Specialist at the Hawaii Department of Agriculture Plant Quarantine Branch. Today, I have the following requests to, 1, allow the importation of 4 African black-footed penguins, Spaniscus dermus, oh, I had it right before, so Spaniscus demersus, an animal on the list of restricted animals Part B by permit for exhibition by Hyatt Regency Maui Resort and Spa. And 2, update permit conditions for the importation of 4 African black-footed penguins, an animal on the list of restricted animals Part B for exhibition by the Hyatt Regency Maui Resort and Spa. Please refer to the first PQB note located on page number 2. The PQB has previously approved import permits for the Hyatt Regency Maui on November 10, 1992 and December 1st, 1992. For black-footed penguins, and then and then the other penguins. You guys can refer to attachments 1 and 2 for those previous import permit conditions and permits. The African black-footed penguins are on the list of restricted animals Part B. Pursuant to the Hawaii Administrative Rules, HAR Chapter 4-71, all species in the family Spinicidae are demersus may be imported into Hawaii for government use or private and commercial use, including research, zoological parks, or aquaculture production. The project background states that the Hyatt Regency Resorts and Spa are can no longer provide viable offspring as they are too closely related, and are seeking to obtain 4 penguins of a different bloodline to ensure genetics vitality. Please refer to the second PQB note located on page 3, which states the Hyatt Regency Maui Resort and Spa has applied for a captive bred wildlife registration, Endangered Species Act, ESA, from the United States Fish and Wildlife Service on 11/10/2021. If this request is approved, PQB would obtain the required license prior to the issuance of an import permit. Currently, they are still waiting on this, and Povi will be able to explain a little bit more later. The objective of this submittal is to obtain 4 new penguins in order to comply with AZA's minimum population standard recommendation of 10 penguins for animal enclosure of our size. And with the introduction of a new bloodline, it is their hope that their penguins will provide healthy offspring to continue the future of their colony. The Hyatt Regency Maui Resort and Spa have had a very successful colony of penguins at the Hyatt Regency Maui for 36 years. Okay, moving on. I have received a total of 4 advisory subcommittee recommendations, and from the recommendations that I have received, 3 of the advisory committee members recommended approval, and 1 advisory subcommittee member also recommended approval. But my approval is conditional on the importer providing definitive evidence that the penguins are free from internal or external parasites. please refer to the third PQB note located on page 11, which states, refer to attachment 8 and 9 for the published research papers provided for review by Dr. Reed demonstrating the known risks for these species. Excuse me, Dr. Maeda was consulted on the information presented. So the, excuse me, so the recommendations was to allow the importation of the 4 African black-footed penguins, an animal on the list of restricted animals Part B by permit for exhibition by the Hyatt Regency Maui Resort and Spa. Okay, for question number 2, it was the same results. 3 of the advisory subcommittee members recommended approval, and 1 advisory subcommittee member stated that, you know, approval is conditional on providing evidence that these animals are free of parasites as discussed above. And the recommendations were to approve and to update permit conditions for the importation of 4 African black-footed penguins, an animal on the list of the restricted animals Part B by permit for exhibition by the Hyatt Regency Maui Resort and Spa. Please refer to the next PQB note 4 and 5, which is located page 13. PQB note states condition number 14A was amended as a result of comments made by Dr. Maeda. He was consulted on the language of this condition as presented. And then the next PQB note, condition number 14B, was inserted as a result of comments and correspondence that was made with Dr. Maeda. He was also consulted on the language of this condition as presented. Okay. Please note that a post-entry inspection was conducted on May 10th, 2022, and there were no deficiencies found during the inspection per the report. Please also note that the Hyatt Regency Maui Resort and Spa has a biosecurity manual on file per permit condition number 17. Thank you, advisory committee members, for your time. This concludes my section of the requests. PQB requests that the advisory committee member or requests your guys' recommendations and comments. Please also note that Povi Carissa Abney is the wildlife supervisor for the Hyatt and is available to answer anybody's questions that they may have. Thank you.
Thank you, Noni. Committee, do I have any questions or concerns for Ms. Putnam or the applicant?
I have one point of clarification on those 2 last PQB notes that were referred to on page 13 that spoke to how conditions were amended or added, Part B, A and B. The notes refer to condition number 14, but it looks like it's condition number 12. I just want to make sure that it's correct or clear what's going on.
That was a mistake on my side. I apologize. That is correct. It's going to be 12A and 12B, not 14A and 14B as listed in the submittal. Apologize for that. Thank you.
Rob, you have any questions or concerns?
I do.
Okay, go ahead.
Maybe we can start with the applicant.
Yes,
yeah.
I'm here.
Hi, Povi, could you state your name and affiliation for the record?
Sure. Can you hear me okay?
We can hear you.
My name is Povi Carissa Abney. I'm the wildlife supervisor here at the Hyatt Regency.
Yeah, I was just wondering if you could tell us what the origin of the birds is.
Absolutely. So we had 3 original penguins that we received from International Animal Exchange about 37 years ago. Those were our 3 founding penguins. One of them lived to 25, one lived to 27, and one lived to age 36. And then they had 6 offspring between the 3 of them, and those are our current residents.
I guess, let me clarify, what will be the origin of the imported penguins that this permit is covered? Where are the penguins that you're going to bring into Hawaii? Where do they come from? Because the application doesn't say anything about the origin of the imported animals.
So we're not 100% sure at this time. Any facility that we're speaking to, we basically have to have the permit in place before they're willing to take the next step. We've already reached out to International Animal Exchange, which is where we got our original 3 penguins. And then we're also in contact with a facility in Florida, as well as SeaWorld in San Diego. Both facilities have an excess number of penguins that were born in captivity.
And then I guess, I hate to ask, but when we weigh these issues, we often look at the risks, which can be difficult to really define in some cases, but weigh the risks against the benefits of potential imports. And I'm just wondering whether you could address what the benefits of bringing these penguins into the state would be for the state of Hawaii.
Absolutely. So when we first received the penguins 36 years ago, they were not considered to be an endangered species. Since then, their wild population has taken a really big nosedive, and it's a fantastic opportunity as far as education and conservation goes with our guests, to be able to explain what issues are happening outside Hawaii, outside our facility, and just give the opportunity to let people know, steps that we can do to help wild populations, not just here in Hawaii, but around the world. It's our goal to keep this colony healthy, to keep generations to come. And we also, just for the mental and social well-being of the animals we currently have, we want them to be able to live full lives. So a lot of the penguins that we have are trying very hard to be parents, but because they're all too closely related, they just don't have that opportunity. So we want to make sure that, not just for our guests and educational opportunities, but for the welfare of our penguins, that they have the fullest life they can possibly have.
Okay, so it has come up in other applications regarding, sanctuaries for animals that might be threatened. And, the question is, well, why can't they be protected somewhere else? Is Hawaii an appropriate place to be protecting colonies of endangered penguins?
So because we have such a similar landscape to their colonies back in South Africa, it does provide a really perfect habitat for them. So their average lifespan in the wild is about 15 to 20 years, and we've been able to keep penguins for 36 years. So quality of life for all 3 of the penguins that we had was well past their life expectancy. We're also able to contribute as far as science and research goes, because we do work so closely with AZA and with other facilities. This is an opportunity for us to supply what we've learned, and if we can contribute as far as population goes, that would be something we would be very proud to do.
And does your education entail explaining to people that penguins are not native to Hawaii and that native bird species typically found in these habitats are extinct or endangered? And what do you do to teach about the native species?
We do, and it is a great opportunity for us to highlight just how delicate our own ecosystem in Hawaii is, by talking about how delicate their system is in South Africa. So it is kind of a full circle moment for us, and just because it's such an unexpected animal to see in Hawaii, it does gain a lot of interest. We have a lot of people that come up and ask questions. It's a really great opportunity for us to interact with people and have them be able to ask and get answers to questions like this. Because it's a fairly small facility, again, it's very accessible, and you know, it's easy for people to come up and approach us and get answers to their questions.
And I guess finally, I'd like to ask about the practicability of testing these birds for avian malaria following the suggested suggestions from the committee as far as multiple PCR tests to ensure that the birds aren't infected, and also a prophylactic treatment. Is that something that's practical?
Absolutely. So yeah, reading Dr. Reed's questions, I think they're very good questions. So this is something that we could have the facility, you know, before they're shipped out, test. We have our own on-staff veterinarian. Well, I guess he's contracted. If this is something that you would like yearly reports, we could continue the testing. Our goal is to be completely compliant. Of course, we want to protect our penguins, but we would also want to protect any birds, any outside animals in the facility.
That's all I had for this.
Okay.
I did have some questions for Noni, but if other committee members have questions for the applicant, I don't want to.
Do we have any other questions for the applicant? Then proceed, Rob, with your questions for Noni. I have some questions for Noni also afterward.
I'm curious as to whether, you mentioned this is restricted use B for research, zoological parks. Is there a definition or requirement for a facility to be considered a zoo?
Not that I know of, but they're also pursuant to the Hawaii Administrative Rules, Chapter HAR, Chapter 471, they can be for private and commercial use as well.
Okay. Thanks, Noni.
Do we have any other questions or concerns from the committee? If not, then I'll ask my questions. Noni, it is clear from the applicant the intent is to breed the penguins. Is this consistent with what PQ is forwarding? Since I see some issues with the permit conditions that would make it difficult for them to breed.
They're bringing it in to breed, that's correct. In the conditions, which conditions are you referring to?
Permit conditions 6 and 12. And it appears that importation and entrance are being used interchangeably, where importation is by definition from 150A-2 shipment to the state from any point outside of the state. And this becomes problematic because permit condition 12 is basically stating any progeny is required to follow any of the pre and post entry requirements for the adult. So I don't see how a progeny can actually be checked 10 days in advance of being imported into the state. This needs to be cleared up, and I think the simplest way to clear it up is by basically stating that the progeny have a different set of permit conditions that apply to them. That they do not have to adhere to the same pre-screening requirements, pre-entry requirements that the adults have to undergo.
That makes sense. Maybe after I speak, Jonathan, if Jonathan has anything, he can chime in. But normally the conditions are for the animals coming in and the progeny, but we could try to make a condition that is, I'm sorry.
Well, I mean, I don't see how you can engage in health checks before importation if importation already occurred and they're being born within the Hyatt's facility.
Yes, so we have the import conditions, but I would also recommend possibly adding another one which talks about just the progeny or so that would be my recommendation.
And keep in mind, these are birds, right? So you have eggs, which you can't do certain things, like you can't mark an egg that will, with an individualized marker that will stay with it since you're marking the shell.
Okay, so with that being said,
that's permit condition 8. And I'm also wondering why we're limiting the language to marking when my understanding is typically birds are banded to give them a unique identifier.
So yeah, the condition is marked with a unique identification code that is approved by the PQB chief. So yeah,
does that include
have different markings such as microchip, bands, rings. There's different types of identifications that's approved for different birds.
Okay.
With that, I'm thinking it would be best if we have add another condition, which is strictly in the event that there is progeny, this is the steps that they would need to follow. Is that correct?
Yes, to me that makes the most sense because a lot of the screening requirements, I don't see how, you know, if the parents don't have any health issues aside from genetic issues, what would the progeny have? If our screening processes are appropriate and are thorough enough, the progeny should not have any zoonotic diseases or issues that should be communicated to them. Unless my understanding of science is wrong and I need to, well, yes, Jonathan.
Jonathan Ho, Inspection and Compliance Section Chief. So historically what we've done is try to have the import conditions manage the progeny as opposed to creating 2 sets of conditions, because the 2 sets of conditions are historically used for transferring, as opposed to having in one facility have 2 sets of conditions, one for the imported animals and one for the progeny. And you are correct in that condition 12 does seem to conflict with reality in terms of, obviously the animals are here beforehand. And obviously, to comply with a 10-day treatment or a 10-day screening process prior to entry to states is impossible. And the intent, I think, is for the restricted animal or the restricted articles, which are the imported ones. And then the progeny really aren't subjected to this because that is detailed with possession in of itself. And I think the striking of progeny, I think makes it very clear that this is for imported animals, not for those that are possessed. And again, and then the idea of the the banding and stuff like that again, and the issue with eggs, the idea of the potential movement of eggs is there for any bird species, not just this one. And this requirement here is really gauged for obviously the adults. And obviously if you're having unbanded birds somewhere that popped up that did not have this mark obviously they had to have either been smuggled into the state or illegally transferred from a permittee that has the birds, and we have an inventory, so we could follow up on that. And again, the intent ultimately is for the permittee to not do illegal transfer or whatever, and if the birds are stolen, obviously that's a completely different issue, but the intent is for them to import, maintain, they cannot transfer, they can't give them away, at least within the state. If they wanted to, they would obviously have to come before the board and go through the process to do that. So I think, yeah, I get where you're coming from, because that does make sense. And I think we're going to have to go through the conditions, because the term progeny is used throughout these conditions. And, if there are clear conflicts for the importation versus the possession, we can make those corrections before going to the board. That's not a problem.
So relating to the eggs, is there a requirement in anywhere, whether it's the SOPs, the biosecurity plan, anywhere, that the applicant has to notify, how do you track the eggs then, since eggs can walk away? Granted, they need to be incubated and everything, but hey, if I know the temperature I need to keep them and how long and how often I need to clean them, it can be done.
Yeah, so the conditions do include number 21, they submit a semi-annual report, including restricted articles as progeny, and they submit the permit number, quantity, and all this kind of stuff. So they do update us with semi-reports, and I have been working with my post-entry sites, and normally if there's some type of birth or some type of death, I'm notified through email regarding it, which goes to their records.
What defines birth for a penguin, Noni? Laying of the egg or hatching of the egg?
I would say if there is eggs available, or if there is egg present, that's a notification. They would notify me, hey, we have eggs over, they're, inform me, hey, we got this many eggs. And then we would just monitor it until they hatch. And then once they hatch, I think Povi was saying that normally when they're born, they have unique spot patterns that help to identify them as well.
Povi, you had a comment?
Yes, so if I could make a recommendation, I was just starting to type it. I would recommend after hatching as being when a penguin would be counted, just simply because currently we've had penguins lay eggs that were not fertile. And that really has to do with how closely that our penguins are related. So if possible, we'll usually try to take an egg and candle it just to see if there's any life inside. And there's also the possibility, again, that some of the eggs don't reach maturity. They never get to a point where they can hatch. But once they are hatched, then that would be a totally different story. Usually what we've done in the past, and this was, it's been 12 years since we've had a baby hatch, we've left it up to parental care, just simply because the babies will be better accepted in the colony if the parents raise it, rather than if we incubate the eggs and raise them separately. If the eggs were getting abandoned, if there were no parental care, then at that point we would generally step in, but that's not something that we've had to do in the past. So as far as marking eggs, removing eggs, it is something that stresses the parents and something that we would try not to do. But generally they only lay 2 eggs at a time, so it's pretty easy to track. And as the eggs get dirty, they'll have certain unique markings, and they generally stay in the same place in the nest. So it is something that we can keep an eye on and differentiate.
Okay. Ken, I know you've had some audio issues. Do you have any questions for either Ms. Putnam or the applicant?
Yeah, number one, are your birds currently banded, and have you had infection problems related to the ban?
We do not currently ban our penguins because they have identifying spot markers. So that's how we can differentiate them. They're very distinct. So it is really easy for us to tell them apart. There are other facilities that banned their birds that have had, I guess, different successes, but generally their colonies, when they ban them, are 40, 50 animals. So it is harder to differentiate. We only have 6.
Yeah, our observation is when birds' legs are banded like canaries, our rate of infection at that point of banding seems to be higher, so that I'm not sure if this would necessarily be appropriate for an endangered species to endanger it more by requiring banding.
I do agree with that. So the majority of our birds, outside the penguins, our swans, our parrots, we've kind of made that same choice not to band, just because we don't want to add any risk to their mobility, to their health, anything like that.
Another couple of points. I don't agree with the contention that the penguins will starve to death if they ever escape. As a practical matter, penguins eat squid and crustaceans as well, and we do have those here. I might also add that I can understand why cold water fish survive better in cold water because of the increased level of long chain omega-3s, but I don't see where the penguin would necessarily suffer from that same problem because they're not in the cold water here.
I'm not sure if that question was directed at me.
It is sort of, because you're alleging that they're going to starve to death because they only eat cold water fish, and the documentation shows that they eat squid and crustaceans as well. Plus, I kind of doubt that they only will eat cold water fish. If given a choice, my guess is they will survive on local fish.
Well, if I can answer that. So every penguin that we have has been born at the Hyatt. They've only been fed a certain number of fish, mainly capelin, and they've never been introduced to squid or any of the fish that we have available, and they've never gone after live bait. So I don't believe they would survive in the wild, just based on their habits. We do have live mosquito fish that are in their water. They'll chase them from time to time, but they'll never eat them. We've tried introducing several different kinds of fish just for a little bit of variety, and they're extremely picky. So we've tried silver sides, we've tried small herring, we've tried other kinds of fish, and they just simply refused it. So that's just based on our 6 penguins and their personalities, and they are a bit spoiled. So they've never really had the opportunity to swim in our oceans and forage on their own. But just having worked with dolphins that have given those same opportunities, they don't readily adapt to foraging on their own and trying new fish.
So your 6 penguins are very human friendly, it sounds like. Are the new imports going to be similarly imprinted?
I would imagine, because all of the facilities we're talking to are, I guess, similar in nature, where they do education programs, where they do training programs. The training programs are really helpful as far as just taking care of the animals without stressing them out. So, we would never even consider taking anything from the wild, just especially since their numbers are so low, and that just wouldn't be a factor for us.
So if they escape, they're not as likely to be hard to recapture because they're going to be friendly to their handlers.
Correct. So we very rarely take them out of their habitat. During shutdown, it is something, we took them out of their habitat to walk them around the lobby. They were incredibly uncomfortable. So they are very familiar with their surroundings. They're not animals that really try to escape. Everything they want is inside their habitat. So their friends, their homes, their food, it's where they're the most comfortable. They tend to be a fairly skittish animal. So even outside the habitat, they do more of like a freeze than a flight. So they're not animals that startle. They're not animals that run. They just, when they're nervous, they just stop. So I don't see that as an issue, especially with the number of people that we have at our facility 24 hours a day. We do have over 150 security cameras. We have staff that are literally 10 steps away from the enclosure at all times. We have security that, monitor the entire property 24 hours a day. So it's not an issue that we've had in the 37 years of keeping penguins. So, I am confident that we can keep them safely.
Would the state accept the pictures of the birds with their unique markings as an alternative to leg banding ID?
Will the state accept that? I'm not sure if I'm the best person to answer that.
No, this
but
would be for Noni, probably.
Okay.
Or Jonathan.
Can you guys hear me? Sorry.
Yeah.
Oh, specific, that would be okay, yes.
I think it's preferable because we don't want an infection problem because we're banning these birds when they're endangered.
Noni, are there other other organizations that have the same species of penguin?
Off the top of my head, there may be 2 facilities that possibly have the same species of penguins.
Okay.
Honolulu Zoo has the same species. Sea Life Park has a different species.
Thank you for the clarification. Do I have any other questions or concerns? Any comments from the public? We received no testimony, submitted testimony. Hearing no comments or concerns from the public, do I have a motion?
I don't necessarily want to make a motion, but would say that I would support this with reservations. My main concern here, I guess I should start out by saying, is the avian malaria. If we added a condition that incorporated Dr. Atkinson's recommendations for the testing and prophylactic treatment, I would support a recommendation with reservations, I think.
Don't we have avian malaria already here?
Correct.
Yes, unfortunately we do. It's a matter of strains and different genotypes. I don't think there would any, they mentioned the mosquito fish. I don't think that's really a sufficient way to guarantee that transmission wouldn't happen if they had an infection to another bird species. So that would eventually, through the chain, come into interaction with native species, which,
I think wild birds would probably fly in, for instance.
Right.
Yeah. So Rob, you would like a modification to, or an addition to permit condition 12 then?
Yeah, I guess wherever it fits in best, wherever PQ staff thinks it would fit, probably 12.
Dr. Hoff, I just wanted to verify. So you're stating to include avian malaria and then prophylactic treatment, was that correct?
Correct, and multiple testing on multiple days of the birds as recommended by Dr. Atkinson.
Thank you.
Hi, Darcy. Povi made a comment.
Oh, okay. Okay, Colby, would you like to speak?
Certainly. So yeah, I just want to support your testing program on behalf of the Hyatt. So whatever testing program you feel we should put into place, we are more than happy to make those modifications. We can continue our penguins and any other birds you feel might be at risk on a regular testing program. We do have a vet who's fantastic, and you know, any testing you feel would help not just our population, but of course Hawaiian populations as a whole, we're 100% on board with.
Am I to interpret that your macaw in the background also supports the conditions?
Yeah, she's laughing right now. So that's yeah, that's R2D2. That's our African gray. And
oh, she's an African gray. R2D2. Okay.
Yeah, she's actually flirting with you, I think, right now. She loves men, so she's giving you a wolf whistle right now, and she's laughing. But yeah, but again, our goal is to whatever standards you put into place, we will 100% follow. So we want to keep our birds safe. We want to keep all of Hawaii's birds safe.
Rob, I do have a question on avian malaria. Is the sampling concentrated in certain areas of the bird? For instance, in Salmonella, seems to be concentrated mostly in the joints between the bones.
I'm not an avian malaria expert, but I believe there are issues with finding the disease in the blood. So that's why you do the repeated testing plus the prophylactic. But I can't answer your question, Ken. I'm a forester.
Okay. Yeah, that's not my area of expertise as well. I just know that in testing for chickens, they can miss it if they're just testing the blood, because it tends to, I should say, most often be found in the joints between the bones.
Okay.
I just want to add that I'll be working with Dr. Maeda and in our office regarding the wording that we will include as a condition prior to going to the presenting it to the board.
Okay. So no brave soul to make a motion. Okay. Then I will.
We're gonna bring back Ken Redmond.
I will channel Ken Redmond to the best of my abilities. I will recommend approval of the submittal contingent upon modifications of the permit conditions to reflect creation of separate permit conditions for progeny, cleaning up of the language throughout the permit conditions, and the use of progeny to indicate that progeny will be held to different standards than the imported articles. Changes to permit condition 12 to encompass avian malaria testing, and the housekeeping errors that Mr. Eisen noted relating to the permit condition. Is that sufficient
Also the banding
detail?
ID.
Oh, and recording of the markings as opposed to banding is if needed.
And Darcy, did you include the prophylactic treatment along with the testing?
Oh, and the prophylactic treatments. Thank you, Rob, for the addition. Clearly, I'm not as good as Ken at capturing all of these in one shot.
That's why we're all too cowardly.
I'm an amateur at recommending motions. Is that sufficient detail for you, Noni? Okay, do I have a second?
I'll second.
Ken seconded. Any discussions on the motion?
I'd just like to say, you know, I find your educational goals admirable, but I question whether, you know, little Tommy from the Midwest who comes to Hawaii on a vacation and returns home, and his teacher asks him what was his favorite thing about Hawaii, and he says, penguins, I saw penguins in the resort. I feel like there is sort of an erasure of sense of place by this, and our authority here isn't to regulate tourism. It seems like this is in alignment with HDOA's rules and protections of the environment. But yeah, I still find this a little bit concerning.
Noted. Thank you. Any other comments or questions? Hearing none, I'll call for the vote. Chair votes aye. Dr. Hawes,
Aye.
Grace,
Aye.
Tom,
Aye.
Rob,
Aye.
Ken,
Aye.
motion carries. Let's see, where are we on the agenda now?
Thank you, everyone.
Thank you, Noni. Thank you, Povi. We are at my favorite part of the agenda, adjournment. Do I have a motion to adjourn?
So moved.
Do I have a second? Tom seconds. Anyone on the committee who does not wish us to adjourn? So please say aye if you agree. Aye.
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 By the way, sorry about being disconnected twice. Our phone connection out here in Waipahu sucks.
No worries, Ken. I'm glad we got you were able to get your questions in and you could hear us. Thank you all. Appreciate everyone's time and effort on reviewing and your participation. This meeting is adjourned at 11:24 a.m. Thank you.
Thank you. Bye.
Why can't I find the disconnect?